What if your tattoo was more than just an artistic expression? What if it could show what your body needs, help optimize your wellness and recovery? Or what if it could help save your life before symptoms even appear? This is the vision behind our project, InkSight, reimagining tattoos as biosensing technologies for real-time health insights. InkSight uses a biological ink made of hydrogel-embedded sensory cells containing pigment-producing compartments. The engineered cells act as an optical biosensor detecting biomarkers in the body's interstitial fluid. In our system, detection of specific targets result in a contrast change visible on the skin. We want to introduce you to three key components that we have engineered for our system, namely the pigment-producing enzymes, the compartments containing the pigment, and finally the receptor that triggers the entire signaling cascade. Pigments represent a class of biopigments acting as molecules for photoprotection, pigmentation, and stress resistance. For our project, we primarily focused on the brown-black pigment eumelanin due to its low toxicity, good visibility, and its relatively simple biosynthesis pathway dependent on a single catalytic enzyme, tyrosinase. While melanin itself is mainly inert, its precursors have cytotoxic and mutagenic properties. In order to not expose ourselves to that, we decided to leverage synthetic compartments within the cells to localize melanin synthesis. In order to further reduce toxicity due to background activity, we engineered different versions of tyrosinases to better control their activity. For that, we developed a tyrosinase that can be activated by TEF protease by modifying the linker region of the self-inhibiting tyrosinase from H and VS, a tyrosinase suggested to us by Dr. Pretzler. We replaced the wild-type linker region with a flexible linker containing a TEF protease cleavable site, allowing directed removal of the inhibiting domain, while also preventing unwanted self-activation by autoproteolysis.
We modeled multiple recombinant versions of these activatable tyrosinases and finally selected two candidates which we expressed in BR21 bacteria and purified by strep tags. In order to test their activities, we performed a tyrosinase assay where we observed a high increase in activity of recombinant HC tyrosinase upon addition of a TEF protease. Inspired by other split proteins, we then created split variants of a tyrosinase originating from Bacillus megatherium. In split proteins, the N and C terminal fragments remain individually inactive, but retain the ability to reconstitute the fully functional enzyme when brought into proximity. Hence, once reassembled, the tyrosinase catalyzes melanin synthesis. Using structure prediction tools, we confirmed the proper reconstitution of the active site and proceeded to express the most promising candidates. However, due to the low molecular weight of the fragments, purification proved challenging. Despite this, we observed a slight melanin production increase when the two fragments were combined, indicating functionality, but further work is also needed to characterize this system. To ensure compartmentalized synthesis of melanin, we decided to make use of the favorable properties of bacterial encapsulins QT, TM, and MX. These proteins self-assemble into nanocages of varying sizes with pores that allow for influx of tyrosine while preventing the efflux of melanin. The encapsulation of specific proteins is mediated by encapsulation tags of the proteins of interest. As recommended to us by Professor Frank, we expressed a variety of engineered encapsulins in HEK cells and tested their correct assembly on native page gels. Firstly, we confirmed that encapsulation signals could be employed to target recombinant tyrosinases to the nanocages. Additionally, we showed that the encapsulins N and C termini could be modified to include functional domains such as external C-terminal fluorescent proteins to simplify experimental design, internally facing N-terminal steric hindering domains to prevent correct assembly until cleavage by TEF protease, and internally facing N-terminal fully functional tyrosinases. To create a tattoo with a change in pigmentation visible to the eye, the required concentration of melanin is crucial. To calculate dopaquinone formation relative to environmental parameters and kinetic properties of tyrosinases, we developed a tyrosinase kinetics model. Under the assumptions that dopaquinone autocatalyzes to form melanin polymers and concentrations are equal, our models predict the concentration of melanin in our cells. Under ideal conditions, a single hydrogel bead, 400 micrometers in diameter, reduces light transmission by 3%.
Our objective was to develop a diagnostic tool as a platform capable of detecting not one, but a range of different targets, while requiring only minimal adaptations to the overall system. To achieve this, we're using a modular extracellular sensor architecture, or MESA. As we have discussed with Professor Leonard, the receptor is highly modular, not only through exchangeable binding domains, like nanobodies, but also via tunable linkers, transmembrane segments, and effector modules. MESA doesn't interfere with the body's natural signaling, and it's designed to detect soluble ligands, making it ideal for our biosensor design. These are the structural elements: 2 transmembrane chains, each with 3 key parts: a ligand binding domain that detects the target molecule, LBD, a transmembrane domain that mediates membrane anchorage, and an intracellular effector domain that initiates the signaling cascade. When a ligand binds to the receptor, it triggers dimerization of the 2 receptor chains. Their interaction initiates an intracellular signaling cascade, which, in our case, leads to change in contrast in an engineered cell. We tested 2 MESA receptor designs in hack cells, a membrane-anchored version for detecting M-cherry and a soluble version for detecting rapamycin. In both instances, we encountered the same problem. The TTA got imported into the nucleus independently of cleavage by TEF protease and/or the presence of the target molecule. In a meeting with Professor Leonard, we hypothesized that too high expression levels of the TTA-BFP protein lead to ER oversaturation, therefore resulting in increased import in the nucleus. In an additional proof of concept, we tested the MESA setup independently of TTA, with BFP only. Similarly, we were not able to observe a clear transition between the inactive state, where BFP-tagged MESA was supposed to localize at the membrane, and activated MESA, with cleaved cytosolic BFP. These results hinted again at too high expression levels of our constructs, and therefore faulty integration into the membrane. In future work, we need to adapt expression levels of the TTA and explore more relevant ligands like progesterone. Our modular system is designed to be versatile to a range of biological targets across different medical areas. We narrowed down our research based on expert input and our survey results to biomarkers involved in hormone tracking like progesterone, immune and inflammatory diseases like LL37, cancer therapeutics and monitoring like EPO and PIAP, and cardiac stress like troponin as possible targets.
The literature contains over 250 validated MESA receptors over dozens of papers. However, understanding the components and designing one of these MESA receptors can take weeks. This is why we present MESA Designer.
MESA Designer is built on experimentally validated components. We integrate over 10,000 antibody structures, curated transmembrane domains, and protease variants, all backed by published data. We spoke to one of the original MESA creators, Professor Dr. Joshua Leonard, who identified our tool as filling a critical gap in the field and provided us with incredibly valuable feedback on how to further improve its capabilities. Let me show you how it works.
Search for your target, let's say progesterone. The software then retrieves validated antibody structures with affinity data. Then select your binding domains with simple drag and drop. Choose validated transmembrane domains and protease variants. All done. Now production-ready gene bank files optimized for your organism are ready to download. We've also created comprehensive video and PDF guides to help you.
We're validating 2 MESA receptors designed entirely with our tool, a progesterone-responsive receptor and a human IgG1 detector, both using components exactly as the software recommended. These constructs are being tested in HEC293 T cells, confirming that MESA Designer generates functional, production-ready constructs.
The best part? MESA Designer is accessible to everyone. You can try it right now with our web interface or deploy locally using Docker, Python, or a REST API. The future of synthetic receptor design is modular and accessible. With MESA Designer, that future is now.
With preliminary results for all relevant components, we developed 3 mechanisms that ultimately lead to a contrast change. Our most simple but slowest strategy relies on ligand-induced expression of encapsulin tyrosinase fusion proteins. Using split tyrosinases, melanin production only starts after spontaneous assembly of the fragments, and pigment gradually accumulates inside the nanocages, darkening the tattoo. Although this method is not suitable for rapid detection of, for example, troponin, it works well for slow changing biomarkers such as progesterone. Faster detection, as required for monitoring and epotherapies, can be achieved by constitutively expressing all components in an inactive form, namely sterically inhibited encapsulins and lid-inhibited tyrosinases comprising an encapsulation signal. In this instance, receptor dimerization leads to the reconstitution of a functional TEF protease that activates both encapsulins and tyrosinases, thus starting pigment synthesis and nanocage formation. Our fastest strategy relies on constant melanin production from active encapsulin tyrosinase constructs, resulting in a tattoo that appears dark by default. Here, the encapsulins are equipped with external TEF-activable coil-coil agglomeration domains, and upon ligand detection, the mesoreceptor reconstitutes the TEF protease, which activates the agglomeration domain and triggers nanocage clustering. The subsequent concentration of pigment rapidly reduces light absorption, resulting in fast depigmentation of the tattoo, making this agglomeration mechanism particularly promising for urgent diagnostic needs, such as the detection of troponin.
You've seen different examples of how InkSight shaped each component of InkSight. But our approach to human practices went beyond just isolated consultations. It was systematic. I would like to show you our flipped pyramid of diversity. First, diversity of stakeholders. We did not limit ourselves to talking to synbio experts, but instead talked to patients, healthcare practitioners, to the tattoo community and artists, and even governance and regulatory specialists. Second, diversity of methods. Different stakeholders require different formats of interaction. That's why our 4 pillars of human practices are interviews, a survey, a policy analysis, and an art competition. Third, diversity of feedback integration. Feedback not only informs our technical and conceptual development, but also how we communicate our work, and it determines the way with which ethical considerations correct the narrative of pure technical feasibility. Through the 26 star-guided interviews and our online survey reaching 246 respondents, we integrated diverse feedback into our design, engineering, and safety work, from construct optimization and mesodesigner development to the designing of a pharmacologically controlled ICASP9 kill switch as one of many measures to address the prevalent public safety concerns revealed in our survey. In our policy analysis, we examined InkSight's potential regulation as a combined ATMP regarding biosafety, biosecurity, ethical aspects of accessibility, and scenarios of misuse. The art competition was ground for creative collaboration with a broader audience and really allowed us to explore the artistic potential of a tattoo-based project like InkSight.
Our project was built through many forms of support.
We want to thank our PI for enabling our work through guidance and infrastructure. We are especially grateful to our advisors who accompanied us with patience, to the stakeholders who engaged in open dialogue with us, sharing feedback and critique. We owe much of our project's depth. And finally, we recognize the essential support of our academic and industrial sponsors who made this endeavor possible.
This is InkSight, the tattoo that tells you more.